Hello everyone, thanks for being here at the Heart Breakout session. I think this is going to be the best session, not because I'm leading it, but because the heart is definitely the best organ ever. So why are we here? We are here because we are still at we are at the reference stage for creating a healthy heart atlas. Ultimately, I guess most of us are interested in studying heart disease, but to do that, we definitely need to create a reference atlas of the adult healthy heart, as well as would be great to join forces with the heart developmental atlas. I guess we can all learn from each other on whatever type of organ we are studying. So I'm Michela Noseda, I work at Imperial College, and my main focus is studying the adult healthy and diseased heart. Since we're very few people, it might be nice that everyone introduces themselves before we go on. We might need the microphone, so let me just grab this. Are these all gonna work? Okay, pass it around, thank you.
No? Yeah. So I'm Sebastian Pott. I'm from the University of Chicago. And so my interest is obviously because the heart is a great organ, but it's also because sort of I'm developing technologies to actually measure different molecular features in single cells. And so what I'm interested in is sort of like the way in which we can like generate, in particular, this heart atlas, measuring as many informative molecular features as possible.
Hi, I'm Xuegong Zhang from Tsinghua University in Beijing. I'm a bioinformatician, so I'm interested in actually everything. So I'm here to learn what special challenges there may be in the heart, and also we have collaborations with heart people back in Beijing.
Yeah, hello, my name is Sascha Sauer, and next to me is my colleague Magdalena Klim. We are from the Max Delbrück Center in Berlin, and we work heavily at the moment on the aging heart. So we look for intrinsic aging processes, and we use single-cell RNA-seq, but in particular also single-cell DNA methylation analysis. And I think we have developed a quite nice toolbox to look at this kind of variation which occur over life.
Hello, good afternoon. My name is Żana Makiedonska. I work at the European Commission, DG Research and Innovation, Unit Head Innovations. You may know that we are in the process of preparing a few projects for funding. I don't have a project that specifically focuses on the heart, but I thought it's an interesting organ, so that's why I'm here. Thank you.
My name's Mark Ilosua. I'm with Holger Hein. I'm taking notes of this session, and I also think the heart is super interesting to study, so I asked to be here.
My name is Greg Edelfinger. I'm from Montreal, Canada. I'm a pediatric cardiologist by trade and interested in cardiovascular genetics, so we're looking at single-cell sequencing of the developing heart.
Hello, I'm Paul Oakley. I'm from a technology company called Dolomite Bio. We do single-cell and single-nucleus encapsulation of cells. And in particular, I'm here because we have methods of encapsulating larger cells, perhaps larger cardiomyocytes.
Hi, I'm Nick Barkas. I work at the DCP. I'm a computational biologist making pipelines for the DCP. My interest in heart stems from my PhD, where I was working on heart development in the mouse. So I kind of know a bit about the field, and I really want to learn more.
Wolfgang Weninger from the Medical University of Vienna. So we are interested in imaging the heart in terms of pinpointing cells we are harvesting from the heart and have the spatial information preserved during harvesting and try to, let's say, are interested in correlating the information received from the heart with the information from the liver, since we are capable of doing, having a kind of body donor facility where we have body donors available which have both organs and other organs as well. So that's the interest.
Hi, my name is Jelach Tjoblume. I'm at biology, in the biology department at Columbia, and I'm moderating the online session.
Thank you. Hi everyone, my name is Krishna. I am one of the program coordinators at CBTM, and I'm one of the main jobs that I have is actually collecting heart samples from recently deceased organ donors. So if you want to know more about getting tissue, let me know.
Hi, my name is François Aguet. I'm at Broad, and I work mainly on the GTEx project. My interests are across many tissues, but muscular tissues in particular for some other projects as well.
I'm Kristen Ardley. I've been PI of the GTEx project. One of the tissues that we sampled was the heart, and we've been doing some single-cell work, and we're particularly interested in population-based single-cell EQTL work in the heart.
I think we have a very nice mixed audience, and hopefully we can have a very interesting chat. I brought with me, I've been given by DCP some slides that summarize the status quo of HCA for the heart. So if you can see here, these are the number of data sets, projects deposited. And if you can see, the most popular seems to be the hemolymphoid system. And for the heart, there have been 2 different projects deposited, one for the mouse, one for the human. For the human, it's data from Sweden from developing heart. So there is already a good head start with some people depositing data, and hopefully will populate this area of the HCA very, very rapidly. The data, as I said, were deposited in Sweden. We're from SmartSeq2, and in the US is data from the tabula muris for the mouse. So a lot of information already available for people to access. Now, I don't know if you guys have started to look at the questions I drafted for us to discuss. One important question for me, and even today with the talks that we have listened to, is about anatomical precision. What I mean here is how precise do we need to be, and how is precision going to affect our results. I mean, the cardiologists and the biologists that work on the heart are going to be very well aware of the anatomy of the heart, but referring to some of the computational biologists and data analysts that we have in the room, keep in mind that the heart is a very complex organ. It is, yes, one organ, but there are at least 4 chambers that are exposed to different pressures of blood, and they have a very different function amongst them. And within the heart, there are structures with very specified, specific functions. If you think to the 4 valves, if you think the interventricular septum, the papillary muscles that help the valves to open and close. So the complexity is, I think, amongst one of the most interesting of all the organs, because of these functional and anatomical differences. And then if you look even, for example, at the level of the left ventricle, and we talk about often in the field about the free wall of the left ventricle, if you section that, you will see that this is quite a thick piece of tissue, above 1 centimeter in an healthy adult. And you will have cells that change from the epicardium to the endocardium. So one of the aims that we are doing in our project studying the heart is to be sure that we capture cells from one side to the other. But this is not simple if you think, for example, to spatial transcriptomics. Now the 10x kit has areas that are 0.6 millimeters, and that cannot cover in one go the whole thickness of the left ventricle. So we definitely have some specific difficulties and issues to solve within the heart. And I'd like to know what you guys think about anatomical precision. What are people doing within this space? We're trying, in our case, to sample at least 6 regions of the heart for now, but it'd be good to know what other people are doing. And just in regarding, because Krishna is here and some of our samples come from Krishna, it's all, sampling from a specific region is not exactly easy, especially when you are in circumstances like Krishna, where you are in a clinical setting, you have to rush and you got a number of priorities. Likewise, if we think samples that have been banked 10, 15, 20 years ago, how sure can we be of the original source of the tissue, and how much can this influence and create noise? I mean, it's real noise, but how can we interpret this when we then translate to the disease samples? Does anyone would like to comment a little bit on this point?
Yes. So I think that basically for using samples that were harvested during operations or during, let's say, not standardized situations. Because you never know whether there is a tuberculated part partly there, and whether there is a thickness or thinness of this part was harvested. And what's vital, in my opinion, is that the coronary vasculature might influence or will influence the transcriptome of the cells. So you would have to have the information of the cardiovascular system in addition to the myocardium position, let's say.
I think I totally agree. As we said, the complexity is immense. On the other hand, we need to start from somewhere. So the difficulty for me is, how do we move on? So at a certain point, we'll have sequenced a number of samples for the most obvious regions. So in that case, we'll have some level of noise because of the patients or donors in this case, or in the healthy heart, and then we'll have some level of noise because one lab across another one won't be able to all sample exactly in the same area, for example, the left ventricle. So what is the best next priority? Do we need to keep sequencing and characterizing more human beings in a relatively precise area, or is it better to go down and dissect a specific area, or do we need to move on for hypersampling and try to go like 20, 30, 50 regions of the heart? What is the sense of the community?
Yeah, so I think to start, 6 areas are great, but it will not be sufficient at all. It's way too few. It would not take into account the areas that are the most diseased, I mean, which is coronary arteries, which is outside of my personal focus, but still, I think that's, then if we look 5 years probably down the line, then that should be very high on the priority list. And I agree that there is a lot of subcompartments that were left out. I mean, trabecular compact versus non-compact, I mean, things like that. But there's also entire parts of the heart that are left out right now. There is no pericardium. There is no conduction system. There is no valves. So also there, we have a lot of potential for disease-related studies. And so I think 6 locations perfect for right now, perfect to start with. And then I appeal to the funding agencies, maybe also to come up with more international calls, to have countries like Canada, who are very reluctant in the uptake of single-cell RNA sequencing, to convince funding agencies that there is something happening at a global level. Okay, we can chat more about this maybe in the break. But I think that's ideal locations to push this further.
I think, thank you
and
for...
go beyond 6 locations.
Yeah, for
geographically in the heart and on the globe.
Raising this issue. Just bear in mind, if we refer to funding, sometimes in our case, we will get the whole heart. So sometimes, so what we do, we process some of the live cells and we'll freeze and fix the rest. But you really feel like, I have an extremely rare sample, and I cannot afford to sample, to analyze it. But I have the capacity, if it wasn't for the money, to sample 30 areas. Okay?
I mean, already logistically, so I'd be interested. So you're getting a sample. So how many people work on that sample?
We do, the samples that we're getting in London and Imperial College, fortunately we can use it for various technologies, not just for single cell. So that's already an advantage. But when we get the whole heart, we could do much more from the single cell point of view.
So you don't have enough hands to dissect?
It's the money to sequence. So for each heart, I can't, I don't have the funding to do that, but the resources are there. And the resources in terms of, tissue, the tissue resources are there, but the funding is not. So and then with the funding means, yes, more people, and of course the sequencing and everything associated to that.
I think we need to convince funding agencies and reviewers that this is a real issue. So one of the critiques I'm getting repeatedly is about the N, how many samples do you have, you know? Why don't you take 10 healthy hearts from somebody and compare them with 10 diseased hearts? Why don't you do that? Then you get statistical power, you know? And I think we've seen how the technique itself is so powerful. I mean, N equals 2 may suffice to start with.
Absolutely.
I think the most important thing would be to come up firsthand with a kind of coordination system, which could be the coronary arteries or whatever, and then have from the very first comparisons the same position in respect to the, let's say, coronaries, or to some system, because otherwise you don't harvest comparable positions, and you might do different sequencing or receive different sequencing results simply because you managed to touch different positions. I think this is the most important way of doing it.
I cannot agree more, and I think during my very first HCA, which was maybe 4 or 5 sessions ago, I mentioned this, and I think we need to have pathologists involved. We need experts that will allow us exactly will help us to map exactly what we're sequencing in terms of anatomical regions. I mean, Krishna and I had a few discussions, tried to design maps and to try and track. Of course, this is going to always be a descriptive type of approach. It'd be great if we can find a way to quantify this, but that will mean imaging, it would be MRI, PET, and I don't think we are ready for that. But again, talking to the funding agencies, I think going down the line, if we could sample hearts in this case, and be able to map with high resolution whole organ imaging, it would be definitely generating much cleaner and more precise data sets. Yeah, so I think here we all agree precision is important, more regions for the heart in the next probably 2 to 5 years as you mentioned, increase drastically the number of regions. Yeah.
One thing I wanted to point out is that in many other organs there's a lot of discussion about spatial transcriptomics. And of course this would be relevant in the heart, but also hemodynamics would play a role, which would vary from person to person and could alter the gene expression at a local level. So it would be good to include that in a long-term goal.
Yeah, definitely. I think in one of my following questions is what level of clinical metadata should we aim for? I mean, when we are getting healthy hearts, it's mostly a person that has disease, the cause of death, just knowing if it's a circulatory death versus a brain death are going to be fundamental informations. So do we need to move on and set almost a threshold of clinical informations that we need to include to be able to interpret the data?
I would say yes. From the perspective of GTEx, we had obviously deceased hearts, deceased individuals, and we had that cause of death. But we did have a pathologist review every piece of tissue. So we had 2 regions of the heart sampled. They reviewed every piece of tissue. I now have several hundred pieces of tissue sitting in the bank, and they would reject something if it was grossly diseased, but if it was diseased within the spectrum of normal for the age of the individual, that was still considered acceptable. So we have this range, but the actual tissue themselves is deeply characterized at the pathological level, which makes a big difference. And you can go back to that, and there's a block as well, and that level of characterization really helps. I'd also say that in terms of this sampling design, and I'm partial to it because I helped think it through with the gut, that outline that Orit presented, where there's a pilot to start with a few regions to get some consistency across labs and across tissues, kind of like this regional, and then a next phase where it goes deeper to capture all the missing regions, but not in a huge number of donors, and then you want to really capture that that angle, that's the variation across individuals and populations, and there you can step back a bit to fewer regions once you know which of the informative regions you really want, or which of the regions you want to compare for disease. So that idea of that sort of 3-stage design, a pilot, a deep, and then a breadth, is a nice way that I think would work here as well.
I think it's a very good summary of how we should approach, and a very practical approach. Anyone has any comment on this? You guys working on the aging heart? I guess you got a variable there that is infinite and absolutely interesting. Yes. Yeah. Okay, so now I also want to point out to the histology, we are doing the same. We try to do H&E plus a number of other stainings for fibrosis, amyloidosis, sarcoidosis, etc., etc. Now my question to you guys is, how do we transfer this information to the HCA? Is it an essential information? Should we be able to upload images of the histology? Should we do that? Should that be almost a requirement? What is the feeling, and how do we translate that to a numerical value? So the integration of those data, which are mostly descriptive as typical histology, histopathology is, how do we translate that to a number, and how we correlate that to the transcriptomics data, especially in a view of having a transition from the typical histology to molecular pathology, and a molecular pathology that is going to go down at the single cell level. Anyone has any thoughts about this?
So may I suggest there was sometimes from Anton Moorman a triggered project in mice, mouse fetuses basically, where they build a kind of avatar of the heart and managed to somehow find some algorithms building in single sections which were stained by the different groups. Maybe this is a kind of approach for such a solution for such a thing, but it's much more difficult, of course, in the human heart because it's of its size simply. So one has to come up with, I think, very innovative imaging approaches to do this. This is the one thing, and the second thing is that I think also for comparisons it's vital to have this reference system then established, because without this reference system it's impossible to build the information in the correct position and to compare different hearts, because then you have to morph the heart somehow, and this is depending on the quality of your reference system.
Is anyone from the DCP here who could think or comment on how we can transfer descriptive data of the histology type into a numerical evaluation or imaging? Or I'd like to know your side.
So I'm not an expert on this particular part, but what I would say is that some kind of metadata update would be required to be able to accommodate that, but that's something which we keep in mind and which should be possible to accommodate. But I can point you to the right person for further discussion.
That's great. Thank you. So now, what progress have we made in analyzing the heart at the single cell level? I think many of you that are in this room will know that we have a specific challenge for the heart, which is the cardiomyocyte size. Cardiomyocytes can be up to 100 micrometers long in disease. They can be even a bit longer than that. They can be multinucleated. So if we think to one of the slides Aviv showed, talking about spatial, how much we go inside a cell to profile that cell, well, imagine that the cardiomyocytes is probably the size of 10, 20 lymphocytes. So if we only think to spatial transcriptomics, how do we go about that? We're going to probably capture a piece of cardiomyocytes across 2 dots, because if the cardiomyocyte is this long and the diameter of a dot of spatial transcriptomics is 100 micrometers, we're probably fragmenting that cardiomyocyte. So I think there is the spatial, I wouldn't say, it's not a big problem. We just need to think of how to interpret our data and how can we move forward to sequence 1 cardiomyocytes at a time with spatial resolution. And on the other side, when we do our standard single cell approaches, most of the technologies that are used at the moment, which is flow cytometry or 10x, have a limitation in size. Now, there are new technologies in the field that are coming up, and I think Paul Oakley might want to tell us a little bit about your technology that allows to sequence or process, isolate cells that are a bit bigger.
Is that working? Yeah. I think not many biologists that use the 10x understand what's actually happening in the black box, and it's rather a simple device for the moment. If you go back into sort of the theory of microfluidics, you can design a microfluidic chip, a junction device, which will accommodate larger cells and larger particles, which is what we've done. Originally, we took the design from Evan McCosco's publication on DropSeq, and we came up with a solution to that, which was slightly easier to use than Evan's original version, but that was still pretty tough to use. And so we came up with an automated way of doing it, and the size of the chip, the junction where the actual droplet is formed that we chose, was 80 microns. That doesn't necessarily mean that a 100 micron item won't go into it. It depends how it presents itself to the junction, and it's possible to make droplets with an 80 micron junction up to maybe 120, 130 or so diameter. The small amount of work that we've done so far using cardiomyocytes seems to be very promising. We have a sort of a pre-filter on the chip which is 100 microns, and when we look at the chip after the run, we don't find lots of samples stuck at that pre-filter, so it looks very promising. The other sort of characteristics we incorporated into the system to make it maybe a bit more cell-friendly, we have temperature control. We also have the ability to stir the sample at various speeds. We can use buffers other than phosphate albumin buffer, so if people have buffers which are particularly preferential to their sample, we can incorporate those into the system as well. So, and that exists today.
I think it'd be good to know if anyone has already tried different platforms to sequence whole cardiomyocytes. The few experiments that I've done in my lab with my group, I asked the transgenic facility that does in vitro fertilization to suck up a few cardiomyocytes for us. So that's obviously a very low throughput, very precise, very efficient. I knew we had nice, healthy cardiomyocytes, but definitely not applicable to large studies. So it'd be great to try new platforms that will allow to sequence cardiomyocyte as a whole in a high throughput. I think many of us now are resourcing to do single nuclei sequencing, which has immense advantages, especially on frozen samples, banked samples. But we have to recognize that we're sequencing only the nuclei. We're not capturing all the RNA that is in the cell. So we're exploring a territory that for most of us is quite unknown. It's exciting, it's great, but I think it's important that we move on and we try to capture also the RNA that is encoded in the whole cell. Yep.
Actually, I have more a question for, I guess, you or other cardiologists. So for us, we use mostly single-cell nuclei sequencing because, well, it has an obvious advantage. But the other question, I think, is like, for mouse data or for mouse heart, we have seen like you can generate quite nice single-cell suspensions, but how well does that actually work? So I think like a second concern is not only sequencing the intact cardiomyocytes, but then also maintaining sort of the proportions of the original tissue. And as one of the problems I think we have encountered there or within other tissues is that you have a lot of disruption of the cells, and if you know, that might disrupt the representation of cell types within your tissue. And I think that's less a comment, that's more a question of like how relevant that problem might be.
I think you're right. I do think that each protocol that we use to isolate cells is going to bias slightly for something. So if we're doing nuclei, we're going to look at the tissue from a certain perspective. If there is now a protocol that I know of that in the heart will allow to isolate all the cells and leave all the cells happy, no. Not that I know of. I don't know if anyone has such a protocol. When we have to isolate cardiomyocytes, we use one specific protocol that tends to preserve and maintain the cardiomyocytes healthy. When we want to really go deep in the stromal analysis, we'll use a protocol that is basically depleting most of the cardiomyocytes. So in my view, at the moment, to get a complete picture of the cells in the heart, we still need different approaches. But that question is different from maybe what you were alluding to. So if I want a technology to compare at the moment an healthy versus a disease and get a sense of the proportion of the various cell types and how these change between an healthy and a disease, probably nuclei are a good way to go. But if I want to answer a functional question regarding a rare cell type or trajectories or whatever other question I want to ask, is that the best experimental design? Maybe not. So I think it really goes down to understanding what is the question, and it's all the issues that we have heard today. I don't think there is one experiment that will allow us to answer everything. We need to use various experimental designs to answer specific questions. Does that answer a little bit? Yeah. Anyone else would like to comment on this? No, that's good. So in this, it's kind of a follow-up of what we're talking about now, which process to use to manage to acquire and understand as comprehensive as possible a cellular landscape of the heart. So if we just take the myocardium of the left ventricle versus the right ventricle and the atria, we're probably going to be able to use the same protocol to obtain cells from these and have a reasonable comparison because we're applying the same protocol. But what about the valves? What about the coronary arteries? I mean, those will likely require very different protocols to isolate the cells. If not very different, I can tell you they're going to be a little bit different. Maybe we have a question?
Yeah, I just wanted to touch upon a comment made online that they basically point out that they're just, for a recent paper on large particle fluorescence, fax-based high-quality single-cell RNA sequencing of adult cardiomyocytes. Was it not? Okay, from start? Okay, so there was an online mention that there's a new method published in Circulation Research that basically uses large particle fax sorting enabled high quality single cell RNA sequencing for adult cardiomyocytes. So that might be something that people want to check out.
Yes, I think in our institution they just acquired one. They use it to sort parasites, so we need to make sure they clean that up before we analyze our cells. But I think it's definitely an interesting paper and a technology to keep in consideration. So going back to what I was mentioning before, I'm curious if any of you guys have started to look at valves and the root of the aorta, coronary arteries from maybe the same hearts, and if you had to develop very different protocols and how you're integrating the data. Maybe not. Hopefully, in the future, we'll be able to get more money so that from the same heart, we'll be able to do, to include all of these regions, which are essential for understanding disease. Also, I think it'd be nice to discuss and see what is your sense in your experiences of what is the noise of variability across the samples. I don't know how many samples have people started to analyze, and if they're able to collect data from a reasonable homogeneous population, or if you have widely different ages, and it'd be nice to hear from you guys. In our case, we are acquiring samples from adults. As I mentioned before, we have a reasonable ratio between male and females. The age tends to be adults. Young adults, not really, because mostly those hearts will go for transplant. And then it'd be nice also to hear what you think about Dana's comments about harmonization of data and batch differences in your data sets and how you are approaching this.
So maybe a comment about the sampling. So in GTEx, for example, for a significant portion of tissues, the sampling instructions are within about 1 or 2 centimeters of a specific region, and we see a huge variability as a result of that. So I think that's just something, unless we come up with a system where we're trying to perform the sampling even more precisely, there's the heterogeneity that results from this is something we'll have to live with. At the same time, there's also, depending on, even though we're considering largely normal tissues, the percent of fat cells, for example, in a piece of tissue can highly vary between individuals, and that will, of course, also affect the overall processing of that sample. So I think going back to comments that were made earlier, I don't really see this as something we can surmount experimentally, and this is really at the computational level of integration that we'll have to take care of it. And most likely it's by looking at transcriptional programs rather than higher level annotations such as states or types that will best be able to achieve that.
Just to add to that, just a curiosity on that, when you're saying that there's quite a lot of Thank you, Francoise. Just to ask a question to GTEx directly is, in terms of the fact that you're saying there's quite a lot of heterogeneity when you're taking a sample within 1 centimeter or 2 of the region you want to pick, is that down specifically to biological heterogeneity, or is that like physical heterogeneity in terms of the tissue? So if I were to take the same, like a sample from the same donor across 2 different centimeter corners, would you see massive heterogeneity there as well? Or is what you're seeing purely biological?
I think it's largely a mix. And it will depend a lot on the tissue and the region of that tissue as well in terms of just within individual variability. But then there's just simply a lot of biological variability. And then also actually on the processing side. I think even though we can't really do this experiment, but if you took the same sample, process it exactly the same way twice, you'll see some technical heterogeneity.
Yeah, I think that depends also, probably, when you say the reproducibility of the same sample run twice. I don't know how you guys are sampling, but whenever we're sampling the LV, I'm always like, oh gosh, do you take a large sample? So the biggest you take, and then you chop it down, and you go and sequence, you're then in the end sequencing only 5,000 cells. So where were those 5,000 cells? So the smaller the sample, you might be more precise anatomically, but then you're afraid of missing the endocardial cells versus the epicardial. And then when you take everything, as you say, you might be sequencing, and yes, what am I getting? The most frequent cells, the tip of the iceberg, or is completely random somehow? So that comes to another question that is bugging me since a little bit. How important is that we go deeper in the sequencing? So is the variation amongst healthy hearts going to increase the more we go down in sequencing? So is it good to stay somewhat shallow and sequence a lot of cells? Do we need to go deeper in those few donors that we are analyzing? I'm just curious to know if anyone has thought about this or tried to do any experiments. Anyone has a microphone?
So I can't comment a lot from the human samples we've done, but I can comment from mouse samples, both whole hearts as well as genetically manipulated animals, knockout animals, and reporter strains. And I think, like in other tissues, there is a couple of extremely important lowly expressed genes in the heart. Like there are transcription factors that we know that do play a role that must be on at that time, and we're not getting to reliable expression levels with the methods we're using. And that is both on drop-seq and 10x, and depth of sequencing does not change that really. So it's an initial capture synthesis problem, I think. And that would lead us again to other types where a deeper sequencing then really makes sense in the way that we're really capturing more genes and that we're getting good signal-to-noise ratios.
So in other words, your suggestion is to use a number of technologies to...
Yeah, I'm not a bioinformatician, but I can tell you the bioinformatics people generate the graphs about the saturation level that we reach with our sequencing, and there is no meaningful increase of gene detection beyond the level we're sequencing, and still we're not seeing the transcripts that we know are there and that do play a role.
Yeah.
I'm not sure whether this is helpful or not, but might it be a possibility to just harvest larger bits, but from the different layers? So to separate the layers, to separate the endocardium, the myocardium with the compact part, with the tropiculated part, and the epicardium and subepicardial fat and these things, and do therefore larger patches, let's say, of this area, but with every single layer differently.
I think, yeah, it may appeal as an idea. For example, we know that in the transmural depolarization gradient, IRX3 is an important gene, but you're not picking that up. And you're not picking up the gradient either, but we know it's there. I mean, it's been shown in a couple of beautiful studies, and just the expression level of those master genes may be so low. You see troponin, myosin, whatever. You see that all the time. But the master regulators, they're at much lower levels, and we're not getting to a meaningful signal-noise ratio, and compartmentalization of your samples wouldn't solve the problem in my eyes. You will remain with the problem that message may be there only, I don't know, 3 times per cell or 5 times per cell, and you're not picking it up.
Yeah. In your case, were you doing nuclei sequencing or whole cell?
That was whole cell.
Whole cell, yeah.
Yeah, just want to confirm that in my experience, beyond 75% coverage of the library in 10x doesn't really help much recover extra genes or separate populations further. So sequencing more samples beyond that point makes more sense than sequencing the same sample over and over again.
Yeah, or use completely different...
or more samples for other technologies. Yeah, or switch to other technologies in a targeted manner once an initial pass identifying the relevant populations has been made.
Yeah, and it goes back to what is the question you're answering. In your case, if you're looking for the gradient, you might focus on the region where you expect the gradient, sequence more cells with a more expensive approach to capture your gradient. So we go back to what basically we started with. One technology is not going to answer all the questions. And it goes back to the money. Yes, as always. Very good. So then, sorry. Yes. I just wanted to conclude. We have a few questions, just a few minutes. And going back to the integration of the clinical metadata, can we establish, or should we try to establish a best practice when we share the data, for example, on HCA? Are there parameters that are typical or important, particularly important to interpret our heart data sets. I mean, in an ideal world, I love to have even just a cardiac ultrasound of each sample, to have troponin T levels circulating, so a number of parameters, but that is a real ideal scenario.
As many as you can possibly get. But really, really key for these type of data are, we have hundreds of metadata points, and they end up as covariates that we have to exclude from analyses. They also allow us to integrate data and exclude data, and it's more important than you could possibly imagine. Especially where there's not a lot of replicate data, where different labs are producing different things. But the technical metadata is really key. How you processed, what you did, where you sampled, what the ischemic time was, or the lag between, if it's surgical, what that time length was. Knowing those makes a massive difference. Adding on the clinical data as well helps, but sometimes, as you say, that's hard to get. You're not going to get that test data. If you can capture it, do capture it. The pathology is something that you can potentially systematically get across everything, and if you digitize images and the DCP hosts them, then you could actually have the cardiovascular community working on it, have a set of pathologists all review and all add comments and come to a consensus. But that technical metadata, the more you get, the better the analysis and integration is.
So I, again, cannot agree more about this. I'm really convinced that we need to almost force people to gather all of those informations and share them, because otherwise we'll end up scratching a lot of our heads and try to harmonize data that are actually not different because the donor, the patient, has a very different history. So I think as a community, we really need to try and make this effort. I think Krishna knows I bugger every other week. Say, can we have all the possible clinical metadata, plus of course the technical metadata which you acquire in the lab is fundamental. So maybe we can just close here. I hope the HCA and DCP can help us, and it might be good to discuss how best to integrate and facilitate the transfer of all this data. Images from histology is going to be relatively easy, but there's going to be a lot of complex and time for transferring the clinical data into tables that then can go to DCP for integration, because a lot of the clinical data are not in tables. I think Korush, the surgeon that works with Krishna, was saying they get PDFs. So how do I efficiently transfer information from a clinical history into chart, a table? It's really time consuming. So a little bit of effort in trying and help us in that would be good from the HCA as a whole. If there are any questions, otherwise, yep.
Maybe one more comment. Mark Halushka mentions online that he's a practicing cardiovascular pathologist, and he's happy to review any histology related to anybody's projects with respect to the heart for both normal and diseased tissue.
That's brilliant. Thank you very much. In our case, just to make a comment, we have a couple of pathologists on 2 collaborating labs that cross check the samples, so that we have even the samples double checked. It'll be good to know if the more pathologists there are available to help, I think the better it is. In the UK especially, clinical pathologists are really swamped by work, and you need to really find someone that is happy to collaborate in the research, otherwise they just don't have the time. So knowing that someone is available to review and contribute, I think it's really greatly appreciated. So if there are no more questions, I think we can go for a coffee. Thank you.